Hi class, we're going to wrap up chapter 11, which we were just finished talking about graded potentials, and now we're going to start talking about action potentials, the actual moving progression, domino effect, so to speak, of this charge distribution down an axon. Let me switch modes here. There we go. And that's the slide we left off on. Let me click over here. That will get us resituated. Okay, so an action potential on one end, it starts on one end of the neuron. Each neuron is going to receive information in the form of graded potentials. So those graded potentials are being kind of localized on the ends of the dendrites in the cell body. And at the other end, the graded potentials at the axon is going to trigger the release of neurotransmitters. So that graded potential is either going to accumulate enough on the dendrites and cell body to polarize that axon hillock / initial segment enough that the rest of the dominoes can fall into place and that action potential can propagate down the axon, or it's not going to be enough and that neuron's not going to fire. So graded potentials only affect that small area, and it's the accumulation of these graded potentials that will, or possibly will, create an action potential. An action potential is going to be the propagated changes in that membrane potential that affects the entire excitable membrane. So let's go back to different types of channels and potentials. The resting potential is going to depend on the leak channels. They're always open, sodium, potassium just going in their directions as, down their concentration gradient. The graded potentials are going to depend on the chemically gated channels, so whether or not they open and close in response to a localized change in chemical concentration. And the action potentials depend on those voltage gated channels. When the adjacent segment is going to depolarize, that's going to cause the gated channels of the next segment to open and allow depolarization there, and it's kind of this domino effect on down the axon. We'll see how it works. So let's look at the steps in action potential generation. At a resting potential, the voltage-gated sodium and potassium channels are closed. Then we reach this depolarization threshold. This is usually when the stimulus is large enough, when there's enough graded potentials to produce an overall change that reaches threshold. The membrane potential at which those channels open, or our threshold, is around negative 60 millivolts. Now remember, our resting potential is negative 70 for muscles and neurons. We'll focus on neurons for this chapter. So our neurons' resting membrane potential is negative 70. It only has to go up 10 millivolts in order for threshold to be met. So it's a relatively low threshold point. The activation of the sodium channels and rapid depolarization is the next step. So the sodium channels are now open as soon as that threshold is met. And what that means is sodium is going to rush in. Those gated channels open, and now instead of it slowly leaking in, the gated channels are now open, and it's like letting loose the floodgates. Sodium rushes into the cell. And because sodium is a positive ion, and it's only the sodium-potassium pump that's pumping it out, we're going to move from a negative charge of negative 60 to a more positive charge. So our charge is going to increase. Once we reach a positive 30 millivolts, once the cell reaches a positive 30 millivolts, the sodium channels inactivate. The gated sodium channels inactivate and close. The potassium channels are now activated. So potassium channels open. And this is going to cause potassium to leave the cell. Because remember, potassium is in high concentrations in the cell. It's slowly leaking out. But when the potassium channels open, potassium is going to rush out. So those voltage-gated potassium channels open, and that's going to allow our potassium ions to move out. And this is the beginning of what we call repolarization. Potassium channels will eventually close once the membrane potential reaches around negative 70 millivolts. Again, there's kind of an overshoot that happens, and we'll talk about that in a minute, but basically the sodium channels are going to shift from being inactivated to closed. So inactivated means they're incapable of opening. Closed means they're now capable of opening. And this is the same idea as a refractory period. You can't have successive nerve impulses. They have to have this minimal refractory period, just like our muscle cells had. So at first, as soon as those sodium channels close and the potassium channels open, those sodium gated channels are inactivated. Then they become capable of opening again after those sodium channels have closed. So as the membrane potential reaches its normal resting membrane potential, -70, those voltage-gated potassium channels start closing, and potassium continues to leave the cell until all gates are closed. And this causes a brief, small hyperpolarization. So instead of just stopping at -70, the cell dips down to about -90. And so here you see this whole process in action. Resting membrane potential, we're sitting here -70. All of a sudden, we get enough graded potentials where we depolarize the cell enough to get to -60. That causes these gated sodium channels to open. Those gated sodium channels open, and all the sodium can now rush in. It rushes in until we get to about +30. At +30, our sodium channels inactivate and close, and our potassium channels open, and now potassium can rush out. At -90, now our potassium channels are going to shut, our sodium channels are open, and the sodium-potassium pump pumps the rest of the sodium out and the rest of the potassium back in to get to -70 and be back at equilibrium or resting membrane potential. It's not really equilibrium. What I suggest doing is summarizing each of these little pictures. You can maybe print out these slides like this, summarize them, and explain what's happening. Or you can print out the slides individually, see if you can place them in order, try to redraw them. Lots of ways you can use these slides to study. That's why I left them all in here. Not to make a really long PowerPoint for you to print out, but to hopefully help you, give you a study tool. So as I mentioned, there's that bit of refractory period. There's an absolute refractory period and a relative refractory period, kind of like with our muscle cells. The absolute refractory period is the time in which the membrane cannot respond at all. It's incapable of responding. And then the relative refractory period is the time when the membrane can respond as long as the stimulus is strong enough. So here you see this whole puzzle, being put together. We have our resting membrane potential at -70. Enough graded potentials add up to get us a threshold, and now our sodium channels open. As they're opening, that sodium's rushing in, they cannot stay open if another stimulus comes. They're going to shut. They will shut once they reach +30, and they will be inactive until we get back down to around -40, which is about when those potassium channels start closing. So potassium channels open up here at +30, sodium channels shut, and the potassium now rushes in. Sodium is stuck in there, and those potassium channels slowly start closing, and we get enough negative potential in our cell to kind of overshoot or hyperpolarize, and the sodium-potassium pump returns everything to normal. So here, again, all of the pictures putting the whole thing in sequence with the absolute and relative refractory periods. Notice once repolarization has occurred, those sodium channels can then go ahead and reopen if there's a strong enough stimulus. This video is on your Mastering A&P Bioflix, I think it's called. So let's talk about the difference between action potentials and graded potentials. One analogy is kind of like firing a gun. A graded potential is like the pressure on the trigger, whereas the action potential is the actual firing of the gun. I was using an engine analogy before, where I was saying the graded potential is kind of like a spark, and the action potential is like when that fire finally ignites, or when the engine finally ignites. So again, the spark, or just that little pressure on the trigger, isn't the actual action potential. It's not going to send a signal. When there's enough stimulus, when there's enough sparks, the gun will fire or the motor will start or whatever, but it still has to reach that point of no return. So, and that's where I just said the point of no return. It's literally called the all or nothing principle, all or none principle. This is where the stimulus is either going to trigger an action potential or it doesn't. There's no like half action potential. It's either an action potential or it's not. All action potentials are equal. The graded potentials are not, but all action potentials are the same. So if there's enough graded potentials, or the graded potentials are strong enough, an action potential will result. And it doesn't matter how strong that stimulus was, it's still going to happen the same way. So you can pause the video here and go back and answer these 3 questions. So let's talk about how action potentials are propagated. This goes back to the concept of myelinization. An action potential is generated along the initial segment first, and then it propagates to every other section of the axon. The action potential at one site can trigger the action potential at the adjacent site, and so on and so forth, because they're voltage-gated channels. This is called propagation. Once the voltage here gets to be low enough, that's going to trigger the voltage-gated channels here, and so on and so forth, as it propagates down the axon. Well, propagation can occur continuously, like I was just showing you, right? Just one segment after the next segment, or it can be saltatory. Saltatory is jumping. So let's talk about continuous propagation. This is on unmyelinated axons. This action potential appears to move along the axon in very, very, very tiny steps. Each step takes only a millisecond, so it's still very, very quick, but it has to be repeated along the entire axon. So it typically takes a lot longer than saltatory propagation. So here you see this whole segment, everything has to depolarize all at once in order, down the entire axon in order to get propagation all the way down to the axon terminals. Saltatory propagation is on the myelinated axons. Here, it's only the nodes of Ranvier, or those unmyelinated parts that are actually depolarizing. The depolarization occurs here and then jumps to the next node and then jumps to the next node and so on and so forth. So it doesn't have to depolarize at every single step, only at the nodes. It's much faster, and typically the speed of impulse is going to vary with the diameter of the axon. Axons that are very large in diameter are going to have less resistance to ion movement, so it's going to be a faster propagation. Kind of like our muscle fibers. Muscle fibers that were big and thick in diameter were fast fibers. Same idea. Here you see the saltatory propagation. These sodium ions can kind of jump from node to node. Video on saltatory propagation and continuous propagation. You can pause the video here, go back and review as needed. It's a gnat floating around. Let's talk about synapses. So now that we've propagated action potential down our axon, we've arrived at a synapse. Our synapse can be chemical or electrical. The synapse usually involves two neurons. One's called the presynaptic neuron, one's called the postsynaptic neuron. Pretty basic terminology there. The chemical synapse is the most common type of synapse. This is where there's a gap between the cells. This is like the neuromuscular junction, right? It's chemical synapse. It relies on a neurotransmitter like acetylcholine. Synapses that release acetylcholine are called cholinergic synapses. So, events at the cholinergic synapse. This is a summary of the neuromuscular junction, by the way. The axon terminal is depolarized by that arriving action potential. Depolarization is going to activate, in this part we skipped in the neuromuscular junction, voltage-gated calcium channels. So the depolarization at the axon terminals is going to stimulate voltage-gated calcium channels. These voltage-gated calcium channels are going to open, causing calcium to rush into the axon. That calcium rushing in is going to trigger exocytosis of our acetylcholine vesicles. So they're going to leave, go into the synaptic cleft. The calcium ions are removed with the acetylcholine as well, so it's kind of recycled. ACH goes into the synaptic cleft and binds to the chemically gated sodium channels. Those chemically gated sodium channels on the postsynaptic membrane will then open in response to the chemical acetylcholine, and that's going to cause depolarization of the postsynaptic cell. And finally, acetylcholinesterase, ACHE, is waiting in the synaptic cleft for the ACH to be done binding to the sodium channels, and then it will be recycled by ACHE. So here you see the whole chain of events. Our postsynaptic cell, or our presynaptic cell here, depolarizing. Our little vesicles are waiting. That depolarization is going to cause our gated calcium channels to open. Calcium rushes in. With that influx of calcium, our acetylcholine is going to be released. Doesn't really show it in lots of detail here. Sorry about that. That acetylcholine is released and the calcium is released also. And that acetylcholine binds to those sodium receptors. Sodium channels open. Sodium rushes into the postsynaptic cell. ACHE is going to gobble up and reuse and recycle the ACH. Here are the individual pictures. Here's a close-up of what's happening at the cholinergic synapse. Again, you have the action potential coming down, calcium ions diffusing in. The calcium ions are going to basically trigger the ACH to be released. ACH is going to bind to our sodium channels. Sodium channels are going to open. ACHE is going to recycle the choline from acetylcholine, and the whole cycle can start again. Here's the whole sequence broken down into many steps. Again, I suggest maybe putting each of these steps on a note card, or maybe trying to redraw it yourself and recreate this diagram yourself. Always a good way to learn is to redraw and recreate things for yourself, because you're using the other side of your brain for that. Let's talk about synaptic fatigue. This is just like muscle fatigue, except we're talking about neurons here. Synaptic fatigue is normally when the neurotransmitters are reabsorbed and recycled. In synaptic fatigue, our synapse is unable to supply enough acetylcholine to be released. So in other words, that recycling is not happening as quickly as it needs to be. So this is typically after extended stimulation. The demand for that neurotransmitter can't keep up with the supply can't keep up with the demand, as I should say. So synaptic delay is the time lag between the arrival of the action potential and the actual release of the neurotransmitter into the synaptic cleft. Usually just half a millisecond, if that, but the cumulative delays can be considerable. So typically our reflexes and very important neural pathways will include a minimal number of synapses. So in other words, if you have important information that has to jump through 3 different nerve cells, that is going to be delayed by about a second and a half, right? Or a second and a half, half a millisecond, half a millisecond, half a millisecond. So you're going to have a huge delay on your reflex or on that information getting sent into wherever it needs to go or being sent out wherever it needs to go. So your body tries to minimize the synapses for the critical pathways. We also have electrical synapses. Electrical synapses are located in both central nervous system and peripheral nervous system, but they're very rare. They're in parts of the brain, parts of the eye, parts of what's called the ciliary ganglia of the peripheral nervous system. There's no variability in response to the postsynaptic cell. It's a more efficient method of transport and communication, but it's less versatile because it's only an electrical pulse that's being generated. It's not different neurotransmitters. So neurotransmitters can give you a variety of effects on that postsynaptic cell, as we'll look at. So in an electrical synapse, it's just a depolarization on one axon, then causing a depolarization on that postsynaptic cell. It's almost a direct connection through special gap junctions. You can pause the video here, go back and answer these questions. Let's talk about what happens on the postsynaptic cell. So the graded potentials that develop on the postsynaptic cell are in response to that neurotransmitter. They can be excitatory or inhibitory. Excitatory postsynaptic potentials, or EPSPs, are graded depolarizations that are caused by the neurotransmitter arrival. This is basically facilitating the membrane or getting the membrane primed for an action potential. The more facilitation, the less additional stimulus is needed for an action potential. So facilitating is getting that membrane close to threshold. An inhibitory postsynaptic potential, or IPSP, is a graded hyperpolarization. So this is opening a chemically gated sodium channel, making it more difficult to reach threshold. So instead of opening up sodium channels where your membrane potential is going from -70 to -60, in hyperpolarization, you're opening up the potassium channels. Potassium's going to leave the cell, and so our cell's going to go from -70 to -80 to -90. It's going to make it harder to get to threshold. So the membrane is inhibited in an inhibitory postsynaptic potential. You need a larger than normal stimulus to trigger an action potential in this case, because you need several graded potentials to add up. You can also have summation. Just like we had wave summation in muscle cells, you have the same principle here in neurons. This is the integrated effects of several graded potentials. It could be the collective effects of EPSPs and IPSPs. One neuron's telling it, yes, you need to depolarize. One neuron's telling it, no, you need to hyperpolarize. And so whichever one has a greater graded potential is the one that wins. Sometimes there's a net effect of no membrane change at all. So again, here you have facilitation with the EPSP, inhibition with the IPSP, or a summation of nothing if both of them are equal, or it could tip the scale in either direction depending on which impulse is stronger, the IPSP or the EPSP. Let's talk about some information processing. All of this information has to be integrated, processed and responded to by neurons or by other body structures. So a single neuron may be receiving information from thousands of synapses. Some of them are excitatory and some of them are inhibitory. The axon hillock is the point of all of this integration. It's integrating all the stimuli, determining how quickly the action potentials are going to be sent, and whether or not an action potential is going to be sent at all. So it's the area closest to the initial segment where that action potential is generated, and the threshold at the axon hillock is lower than anywhere else in the cell's body. That's its most excitable part. So the simplest level of information processing in the nervous system is when our neuron is changing in response to or responding to changes in like oxygen, nutrients, abnormal chemicals. That would be the easiest way for our cells to relay information when it's something life-threatening like oxygen levels, right? So this is an example of multiple synapses. I mean, look how overwhelmed this poor little guy is, right? All of these axon terminals, that's the purple things coming in. The yellow are the glial, like neuroglial cells, cell processes coming in and either protecting or nourishing that neuron, making sure that it's getting the nutrients that it needs. So let's talk about some types of summation. You can have temporal and spatial summation. Again, summation is when there's a combination of EPSPs, IPSPs, or one or the other that's going to be basically added up on our postsynaptic cell membrane. So temporal summation is when a single synapse is repeatedly activated. This would be when like the excitatory postsynaptic potential is just maximally stimulating that postsynaptic cell. So each new action potential is causing release of more acetylcholine into the synaptic cleft. More ACH equals more depolarization equals the ability to get to threshold quicker. So it's kind of like an emergency type thing. That's temporal summation. Just over and over and over again being stimulated. Spatial summation involves multiple synapses that are active at the same time. So this is a cumulative effect. So the degree of depolarization is going to be determined by the number of excitatory synapses and the distances those synapses are from the initial segment. So the action potential is generated if that membrane reaches threshold. So again, a couple different synapses coming in, hopefully threshold will be reached and that action potential can be generated. These questions here relate to what we just covered. You can pause the video, go back and review them. Let's talk about regulatory neurons. This is higher levels of information processing. Regulatory neurons are either going to facilitate or inhibit the activities of a presynaptic neuron. It's going to affect the cell body, the membrane of the cell body, and the sensitivity of the axon terminal. So they play a huge role, those regulatory neurons. We also have more neurotransmitters. There's more than 100 different neurotransmitters in our body, so they each work in a different way. We've been talking about the cholinergic receptors and their role in just generally communicating with postsynaptic cells, but there's hundreds of neurotransmitters that are in our bodies, and they each have different effects on postsynaptic cells. Some may have direct effects on ion channels specifically. Some may have indirect effects. Some may involve something called a G protein. A G protein is an enzyme complex that's actually attached to the membrane receptor. When something binds or something activates that cell membrane, the G protein is compromised or changed in some way, and it can trigger something called a second messenger. A second messenger is going to alter whatever's going on in the cytoplasm. It could even alter what's going on in the nucleus, depending on what that message is. This chart here is just showing you different neurotransmitters. You're not going to have to recognize their shape, but you should be familiar with the neurotransmitters and their overall mechanism of action. In other words, if they are located in the CNS or PNS and what types of junctions they're related to, what types of interactions they're going to be involved with. So we also have interpretation of highly complex information where information is basically being translated into action potentials and propagated along axons. The message that is arrived is then integrated on the frequency of those arriving action potentials. So muscle contraction is going to change in response to increasing frequency of action potentials. If your motor unit or your motor neuron only fires a few times, your muscle might tense and then relax. But if your motor neurons are continually firing, your muscle may stay contracted or may continue flexing or moving or whatever it needs to do based on that neural input. So again, this is where you get those twitch, contractions, tetanus, incomplete tetanus, all those things that we talked about before, wave summation, etc. This chart just goes through the overall summary of information processing, and here are your 3 review questions for the end of chapter 11. Let me know what questions you have, and I'd be happy to answer them. This was a dense chapter full of neuron information, but I want to say I promise you it gets easier, but it really doesn't. The next couple of chapters on the nervous system are pretty physiology intense, so it's important that you get a really good, strong grasp on the neurons and how they work and how action potentials work, so that you can start building an understanding of the whole picture of the central nervous system and peripheral nervous system in the next few chapters. So again, let me know what questions you have, and I'll see you later. Bye.